This video, let us answer section A of the 2024 GCE Biology paper 2. So we are going to answer section A questions. Okay, and as you can see from this paper, you only have 1 hour 45 minutes. So this one shows how fast you need to be in terms of answering questions. So you need to be quick in terms of answering questions. This paper is comprised of section A and section B. Section A, where you find one word answers or explaining, but section B, you find essay questions where you need to write your work in paragraphs and of course in full sentences. So let us look at the first question that came. So as always, the first question came from cells. Now, as you can see, I'll take time also to explain, maybe just to give you a guide of what might be possible questions that you expect in the internal ones, those who are writing internal in November. Here what came, it was a typical animal cell. And now what you might expect in the November one, you might expect one of the specialized cells. So you need to make sure that you know specialized cells. Okay. So the question is, and by the way, this section gives you 44 marks. So answer all questions in this answer all questions in the space provided in the question paper. The question reads, Figure 1 shows generalized animal cell. Like I said, just a typical animal cell. Identify the parts A, B, and C. Now, this one was simple. 3 marks for free. Identify the parts labeled A, B, C. So what are the parts? So the part A is the cytoplasm. Then of course the part B is the cell membrane. The part C is the nucleus. So these are the parts. So 3 marks for free. Name the organelle present only in plant cell. So what I'm simply asking you here is that of the parts that you have here, which of the 2 are found in plant cell only? So here you can talk of the cell membrane which is B, and of course the nucleus which is C. Okay, so you can put just these 2. But you know also that the cytoplasm is found in plant cell. Okay, then C, explain the function of the part labeled B. The part labeled B is the cell membrane. Now, the part labeled B is the cell membrane. So what is the function of the cell membrane? So the function of the cell membrane is simply to control the movement of substance in and out of the cell. So you can say controls the movement of substances. Control the movement of substances in and out of the cell. Now here you need to be careful, please. As you look at these parts of cells, the adaptation of the cell membrane is that it is selectively permeable, and that is not a function, because some of you might go that way saying, no, the cell membrane, the function is that it's selectively permeable. No, that is just an adaptation for its function, which is controlling the movement of a substance in and out of the cells. And this is based on the size of the substances. So smaller substances or small substances are able to enter the cell without struggle, without being blocked, and of course come out of the cell. But of course large molecules or substances are not able to pass through the cell membrane. There are different other methods by which they are transported, of which we are not going to go into detail into that. Describe the effect of cells if it was placed in hypertonic solution. So before you give the effect, you need to know what is the hypertonic solution in the case you're asked that question. So the hypertonic solution, if you are comparing the concentration of a cell that is inside cell, and of course the solution where you put it, Hypertonic solution is the one that is more concentrated compared to the inside cell. So more concentrated here, which means it has less water. Then the inside cell has more water. Now, if you are to talk of osmosis, you know, it's the movement of water from the dilute solution or from the region of high water potential to the low one. So water will move from the cell to the solution, and therefore the cell, since it's an animal cell, will be crinated or it will shrink. So in writing here, you can say what will happen, the cell will lose water and become crinated. I think this is enough to earn you 2 marks. The cell will lose water due to osmosis. You can add that. The cell will lose water due to osmosis and become what? Crinated. And you know the reason why it will lose water. Because the outside is concentrated compared to the inside one. Okay, so here is the second question that came. And this one was on saprophytic nutrition. And this question is more like a repetition of the question that was there in 2023, if I'm not mistaken. But the questions are different, but the diagram looks familiar, or looks same. So now the first question is identify the part D and G. From this diagram, what is the part D and G? So the part D is the top part there. So that is called, since it's just pointing at one, that is called the sporangium. So that is the sporangium. And of course, the function of the sporangium, just to give here in case you find another question asking you about that, it bears what? It bears spores. So spores in saprophytic nutrition here is more like seeds. They germinate under favorable condition. And the more reason why saprophytic nutrition occur, it is because it might not, the conditions for, by the way, you know that a saprophytic nutrition, okay, let me not talk about that. I wanted to explain about asexual and sexual, but anyway, I'll not go into that. So it bears what? It bears spores. And spores are seed-like, which just germinate when the conditions are favorable. Then, of course, let us look at G. G, as you can see, is the down part there. So that part is called the rhizoid. Okay, so those are rhizoids. Okay, I just put rhizoid. Now, the function of a rhizoid, you know that is also called as the feeding hypha or hypha, however you pronounce it. This one, feeding hypha. So what it does is that at a tip there, it produces extracellular enzymes. You know what happened in saprophytic nutrition, yeah? Enzymes are produced to hydrolyze a substrate, which might be proteins, carbohydrate, fats, and the end products are absorbed. So one, the function of the rhizoid is to secrete extracellular enzymes and also absorb the end product of digestion. So it acts also like a feeding, or it's a feeding hypha. It's known as a feeding hypha. Okay, since we are just told to give the parts, we can end here. Explain the function of the part E and F. Where is the part E? So the part E is this part. Now, before we give the function of the part, you need to know the part. So the part E is the sporangiophore. Okay, I hope I've pronounced it well. The sporangiophore. So for the sporangiophore, the function is simply to support the what? The sporangium at the end. Okay, so the part E is the sporangiophore, and of course we can say supports the what? The sporangium or sporangia. You know that sporangia produce spores. Then what about F? Part F is called the stolon. Part F is called the what? The stolon. And now if I were to look at the stolon, it's the horizontal. It's the horizontal one. Look at this. This horizontal part, that is where the upward part here where these sporangiophyll and the rhizoid that are going down here develop or grows. So now what could be the function of the stolon? Okay, so one of the function is that it grows along the substrate. So one of the function you can say grows along the what? The substrate. Grows along the substrate. One of the function. Then bears the the bears, so we can say bears the what? The sporangiophyll and rhizoids. I know the question is not asking us to do this, but just have to answer the question, even the question that we might expect in the near future. So grows along the substrate, bears the sporangiophyll. The other question we can say it grows along the surface of the substrate to obtain air for respiration. So it grows along the surface of the substrate, and this is the one that obtain air for what? For respiration. So if you can give any of these three functions, one of them, you are right. Okay. Then now the other questions outline the process of nutrition. Outline the process of nutrition carried out by the fungus. Now, fungus is one of the one that carries out saprophytic nutrition. Now they are asking you, oh, I've even just said that, because the kind of nutrition that we are going to outline here is saprophytic nutrition. So outline the process of nutrition carried out by the fungus. You're going to start by identifying the kind of nutrition, and that is called saprophytic nutrition. So we can say, in writing we can say, look at this is 3 marks. So you can say fungus carry out saprophytic what? Nutrition. Now here let us define what saprophytic nutrition is. So we can say saprophytic nutrition is a kind of nutrition. It's a kind of nutrition that involves obtaining nutrients from dead and decaying matter. Okay, decaying matter. So now fungus carry out saprophytic nutrition, and saprophytic nutrition is a kind of nutrition that involves obtaining nutrients from dead and decaying matter. And if you're asked to describe how saprophytic nutrition, or to outline how saprophytic nutrition is brought about, usually in this case saprophytes, in this case the fungus, releases enzymes at the end of, that is, the releases extracellular enzymes at the tip of the rhizoid. The enzyme digests the substrate externally, and of course absorb the nutrients internally, which are used, which are later assimilated, okay. So usually saprophytes releases digestive enzymes at the tip of the rhizoid, which hydrolyze the substrate. So if you are talking of carbohydrates, the end product is glucose. If you are talking of proteins, the end product are amino acids. Lipids, end product fat acid and glycerol. These are later absorbed by the rhizoid. Like I've said, it's also known as the feeding heifer. Then of course is assimilated internally or utilized internally. So that is how it is brought about. And of course I have a recording on the same part. I guess you can, or you might want to check out that one. Then give an example of a fungus that carries out the process of nutrition shown. Give an example of fungus that carries out the process of nutrition shown above. Okay? So 1, you have mucor. I guess this is the correct spelling. Then of course you have also what is known as rhizopus. Rhizopus. Mucor rhizopus. Okay, so we can talk of these as examples. So this brings us to the end of question 2. Let us see question 3. So for the first time, I've seen these diagrams. I think I'm seeing these diagrams in the questions. So now the questions, the way are prepared, please. You need to expect things that you've not seen before. The kind of phrasing of questions that you've not seen before. Okay, so be expectant. So for question 3, figure 3 shows kind of germination in seeds after it was planted in a well-watered soil. So water as the condition required for germination was a variable. Okay, and other conditions in fact. So what characteristics of living organism is demonstrated above? Look at this. We are starting with the seed, coming to the seed stage 2. Here, germination. And of course, you can see the improvement or the growth. So here, the kind of characteristic is called growth. Okay? And I hope you know how to define growth at a moment. Okay? Identify the part H and D. I. So the part H is this part, which is the shoot. But here, where it is still like this is called, the shoot is called the promule, however you pronounce this one. Then of course we have this part I, which is called the radicle. So H is the shoot, the promule. Then of course we have I, which is the radicle. The radicle is the one that develops into roots. Okay? So these roots were radical. Okay, state the kind of germination shown above. Now, this kind of germination shown above, as you can see, look at this. This one is, as you can see, this is a dicot. Okay, it has 2 cotyledons. And dicot, the kind of germination they undergo is called epigeo kind of germination. Okay, so epigeo germination. Okay, why is it epigeo germination? Let us see the question. We might be answering the questions. Let us answer them in order. So state the kind of germination is called epigeo germination. Epigeo germination. Give the reason for your answer. So what is the reason for your answer? So because the cotyledon, I'm going back to the diagram soon, the cotyledons are pushed up by an elongation of hypocotyl. Okay, hypocotyl, something like this. Okay, so look at this. This is the cotyledon, okay, in a tester. As it break out, look at this. These are cotyledons. So they are all pushed out as they germinate. But of course, for the other kind of germination, this remains underground. Then what comes out is just the shoot. I guess you've seen that. Okay, so that is the reason why we are saying it's an epigeo germination. Explain the importance of water in seed germination. So what is the importance of water in seed germination? So one of the importance is that it softens. Okay, so for seed to germinate, as you can see here, it has to break out of the tester. So when it absorbs water, it softens the tester so that so that is the seed can split. So softens the tester, so that it can split. So those that might not be aware, the tester is just the cover outside the seed, okay, to release the promule. Okay, so the promule is the one that develop into a shoot, and the radicle, the one that develop into what? Roots. That is one of the function of water. Okay, or the importance of water. The other one is that it activates the enzymes. Okay, it activates the enzymes. Okay, that is involved now. That activates the enzyme and provides an aqueous medium for metabolic reaction to take place. Okay, so for metabolic reaction to take place, the enzymes inside need to be activated, and water is one of the material that is needed. Okay, so that, or those are some of the importance of water that you can look at. You can also look at it is involved in hydrolyzing of what? Nutrient into disaccharides. Let me talk of carbohydrates in the seed is hydrolyzed to maltose, okay, because of water. So quite a number of functions that you can look at. So please do not be close to what we've just looked at here. Make sure that as you study, you look for some more. Apart from moisture, state 2 other conditions necessary for germination. So one other condition necessary for germination is oxygen. And of course, oxygen is required for respiration. Okay, so we can say required for respiration. Okay, so for respiration to take place here, oxygen is needed. Then also we can talk of suitable temperature. So if I was marking, then you just write temperature, I would surely give you a 0 mark, because we are not just talking of temperature but suitable temperature. You know that here there's there are enzymes involved, and you know that enzymes activity are affected by temperature. So why I'm saying suitable temperature, it has to be the optimum temperature in which that enzyme works the best. Because if the temperature is high, the enzyme will be denatured. If the temperature is low, the enzyme will be inactive. So suitable temperature is needed. I hope I've explained there. So this brings us to the end of question 2. And now question 4. Figure 4 shows the spinal reflex arc. Okay. Identify the parts labeled J and L. Identify the parts labeled J and L. What are the parts labeled J and L? Okay, so this question, please, you need to know that this is coming from nervous system. So the part labeled J, you can put dendrion or receptor. Okay, so let me just put dendrions, dendrites, receptor. Then of course we have L. What about L? Where is L? So L is right there. So this is the white matter. Okay, that is white matter. Then explain the function of part labeled M and O. Explain the parts, the function of the parts labeled M and O. Where is the part M? The part M here. Now, before we give the function, this part, you know that is the muscle. Now, what is the function of the muscle? So the muscle is a receptor. Okay? I mean effector. So the muscle is the effector and its function to carry out response to the what? To carry out response to the stimuli or electric impulse. So carry out response to electric impulse or nerve impulse. That is the function. What about the parts labeled M? I mean the part labeled M. Is it M? Oh, this one. Now this part here is in the middle of, by the way, this is called sensory. This is called sensory nerve. And of course the other one is what? Or sensory neuron. The other one is the motor neuron. Then this in the middle is called interneuron or relay neuron. So interneuron or relay neuron. So the function here, it links the motor neuron and the sensory neuron. Links the sensory neuron and motor neuron. Okay, the other one we can say transmit. So we can say transmit nerve impulse from the central nervous system to the motor neuron, or we can say from the sensory neuron to the motor neuron. Okay. Okay, so I guess you've gotten this part. Then of course now we are saying which letters from figure 4 or in figure 4 forms part of the spinal cord. So which letters in this figure forms part of the spinal cord? It happens that it is K and L. So K and L forms the spinal cord, and K is called the gray matter. Gray matter. Then of course, is it L? We said already is the white matter. So the white matter and the gray matter makes up the what? The spinal cord. Okay. Then what about this circle here? What is this circle here? This in the middle here. Well, that is called a central canal. Central canal. So in the case you're asked in the next part, maybe what is that? Then you're able to tell. Then of course here, that is question D. Give one example of a spinal reflex action. Now, spinal reflex action are those that are just processed within the what? The spinal cord. And in this case, the example of spinal reflex act, we can talk of withdrawing your hand when you touch hot object. Withdrawing hand when you touch hot object. Okay, withdrawing of hand when you touch the hot object. Then, of course, what about cranial reflex? Those that are processed. Okay? The procession happen in the brain. So you can talk of what? Blinking as an example. Okay? What is the overall function of the nervous system? What is the overall function of the nervous system? So the overall function of the nervous system, we can look at respond to stimuli, both internally and externally. It responds to stimuli. Okay? So in writing, let us say we can say send messages from various, or let me just say respond to stimuli. Okay? And this now can be summarized in terms of the functions that it plays for sensation, response, integration, and controlling. Okay? This summarizes the function of the what? The nervous system. So it helps us to respond to stimuli. Stimuli, we are looking at the changes. Okay? It can be the changes in temperature, changes in pressure, and all that, and so on and so forth. Okay? So respond to stimuli. It helps us to respond to stimuli or to changes in the environment. Okay, so please also be ready to look at how you can phrase this or how can you put the answer. But remember, it's just a 1 mark. So this brings us to the end of question 4, and now up to question 5, that is on genetics. So as always, genetics, question 5, section A, you always find the question on genetics. So be prepared for that. And the first question here is, what is mutation? So mutation is a sudden and spontaneous change in the structure. So in the structure of a gene or number of chromosome. I presume that up to this point, or you've done genetics. There are 2 types of mutation: gene mutation and, of course, a chromosome mutation. Then state 2 causes of mutation. So one of the causes of mutation are chemicals. So certain chemicals cause mutation. And of course, we can talk of radiations. Okay, so radiations also cause mutation. So these are the 2 causes of mutation. Then now here we have a couple, that is question B. A couple both with normal skin color produce an albino child. So both with normal skin color produced a child. Now what you should know is that this albinism is expressed, expressed in when you have all the recessive alleles, in short. So when all are recessive, that is when someone becomes or have this condition called albinism. So a couple with normal skin color produced an albino child. So which means these were carriers for albinism. So express using the genetic diagram how this would have been possible using the letters or alleles A. So alleles A and small letter a. You know that allele or capital letter A is for dominant, then small letter is for recessive. So now, if we are to put up the genetic diagram, we are going to start, let us start with parents. Then we are going to put what? Male. And here we're going to put what? Female. Or mother and father. Okay, we can put father. Then we can put what? Mother. Then of course, let us look at parental. Parental phenotype. What were the parental phenotype? So parental phenotype, we are looking at normal, and of course this one also normal. Then parental genotype. What was the genetic makeup? So we have A, capital letter and small letter. A, capital letter and small letter. Then of course we go for parental gametes. So for parental gametes, you know that we put this in circles. Then also this one. Okay. Then now we close. Fertilization occur. Here we have A. Here we have A. Then we have A. So we start with the dominant one. Then now we have A. So we have first generation genotype. So F1 offspring genotype. So these are the genotype. So F1 offspring phenotype. Then we have this one normal, but carrier. Okay, so normal. Also this one what? Normal. This one is the albino. So this is how possible that although they were normal, but they had one child who is albino, as you can see from here. Okay, so this is the genetic diagram. You can even put here fertilization or meiosis. I'm not saying you underline. So this is the genetic diagram, and this brings us to the end of section A questions. So up next is section B, but of course we are going to cover this in a different video. So please, if you enjoyed the lesson, make sure to share with more. Make sure you subscribe to the channel so that you do not miss out for more videos. Otherwise, we can end here. And let me just say, we are doing, we are registering for GCE 2025. So those who want to join us for GCE 2025, you can WhatsApp the number 0976 40 2563. Also, internals, we are doing past paper revisions. If you want to join us for revision, you can still join us. It's just 100 kwacha per subject. Be part of this, be part of the people that you are preparing for the exams. Otherwise, see you in the next one.